Welcome to Let's Talk Micro. Hello, my dear audience. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And if you're listening to this and you celebrate Thanksgiving, happy Thanksgiving. I hope you're having a wonderful time with your family and loved ones or friends or whoever you normally celebrate with. But at least, I hope you're getting some rest and recharging those batteries. And of course, you can always find Let's Talk Micro on Apple Podcasts, Spotify, Amazon Music, GoodPods, wherever you listen to podcasts, you can find Let's Talk Micro. As far as social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I have an email address which is letstalkmicro@outlook.com. So either via social media or via email, you can send any feedback, any topic suggestions. They are always welcome and appreciated. So please go ahead and subscribe to the podcast, download episodes, and if the app allows you to do so, please go ahead and leave a review. Thank you so much for the support. And as you know, the Let's Talk Micro podcast does not have any sponsors, so if you want to help the podcast in any way, you can do so via Venmo or via Buy Me a Coffee, KOFI, and both links are available on the show notes. And of course, if you're looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. And of course, these are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. If you haven't listened to the previous episode of Let's Talk Micro, please go ahead and do so. It was a great episode where Ranim Palota, who's a pharmacist at the University of Utah in Salt Lake City, she joined the podcast. And in the episode, she talked about an article that she co-wrote, and it was about an NDM variant that was seen in E. coli, an NDM5 variant, actually. And of course, we started with an overview of what are beta-lactams, beta-lactamases, penicillin-binding proteins. And one of the beta-lactamases is New Delhi metallobeta-lactamase, or NDM. And those of you that work in micro and microbiologists, pharmacists, ID physicians, you know that NDM actually affects the activity of beta-lactam drugs with the exception of estronam. And in the episode, she talks about this variant and how, it has a PVP alteration and how this affects antibiotics. So it was definitely a great episode, so please check it out if you haven't already. Okay, so let's go ahead and talk about today's episode. So those of you that listen to the podcast regularly, you know that I recently re-released an episode that I had done last year about veterinary microbiology, where Kelli Maddock and Sarah Gerfro, they joined the podcast to talk about what's microbiology like in a veterinary lab. And it was definitely a great episode. So in today's episode, I am joined by Dr. Kelli Maddock. So she returns to the podcast, and she is the section head at the North Dakota State University Veterinary Diagnostic Laboratory. And she came to the podcast to talk about my favorite organism, which is Pasteurella multocida. And if you have been following the podcast for a while, you know that at the beginning in 2021, I did an episode about this organism and why do I love it so much. So in the episode, Kelli starts going over an overview, like a historical overview of Pasteurella multocida, and then she moves on to talk about the capsular types. And in the episode, she also talks about her doctoral project that she did, which the link to that study, it's in the show notes. And in the study, they use MALDI-TOF MS as a rapid way to look for hemorrhagic septicemia types of Pasteurella multocida. And in the episode, she explains how they did that. She talks about hemorrhagic septicemia, and it was such a great episode. I definitely love talking about Pasteurella. I really love that Kelli also, that is her favorite organism. And one interesting fact that she mentions in the episode is actually that multocida means killer of many. So once again, so great having Kelli back. All in all, a great episode, so let's go ahead and listen to it. Well, today is a great day at the Let's Talk Micro podcast. Those of you that have been listening for a while, you know that my favorite organism is Pasteurella. So if you like it too, and maybe if you don't, definitely stay and listen to this episode because it's great. We're going to talk about Pasteurella. So with me today, I have a guest, and she was a guest before, If you haven't enjoyed the episode about veterinary microbiology, I definitely invite you to check it out. But in the episode, I had Kelli Maddock and Sarah Gerfro. So with me today, we have the now Dr. Kelli Maddock. Kelli, welcome to Let's Talk Micro.
Thanks. I'm excited to be here.
My pleasure. And we talked about this off screen before, but once again, it's definitely as someone that's about to finish his master's and I know the hard work that goes into that with a full-time job. You just completed your degree not too long ago, so definitely congratulations.
Thanks.
Okay, so it has been a while since you've been here, and so maybe we can start with a quick introduction. So if someone is just listening and haven't listened to the previous episode, so they have an idea of what you do.
Yeah, I'm Kelli Maddock. I am section head of the Microbiology and Biosafety Level 3 laboratories at the North Dakota State University Veterinary Diagnostic Laboratory. I'm a medical laboratory scientist by training with my bachelor's and master's in medical laboratory science. And as Louise just said, I just finished up with my doctorate in public health from the University of South Florida. Very passionate about One Health and the human-animal interface when it comes to bacteria too.
Yeah, definitely, and I always get good feedback from the episode that we did before. People working in the clinical lab and especially medical lab scientists and seeing things happening on a different population, which in your case is veterinary. So I think it definitely had a lot of good feedback. So let's go ahead and, as I said, we're gonna talk about Pasteurella, more specifically Pasteurella multocida. So maybe let's go ahead and start with a historical overview.
Yeah, so Pasteurella, as you can imagine with the name Pasteur snuck in there, Louis Pasteur had a hand in identifying this bacteria. Of course, disease caused by what we presume would be Pasteurella maltosida at the time had been described before. But Louis Pasteur essentially identified it as the causative agent of fowl cholera. So that's a disease in birds. And also through this, of course, those happy accidents and miscommunications, the culture wound up being left out for a month, and it wound up becoming an attenuated vaccine. So there were some initial vaccine studies done with Pasteurella maltosida. That's about 1879. And over the years, many different animal diseases were described as being caused by this bacillus, as they would call it at the time. Um, and as time went on, other diseases such as hemorrhagic septicemia were described in about the 1880s. Early 1900s wound up with the more of that recognition of the various animal diseases. But then there was also a lot of debate on what was actually causing the disease because they'd find different bacteria, which I think they later figured out was this another Pasteurelliaceae, that Mannheimia hemolytica. Um, and in 1912, again, this hemorrhagic septicemia, we can go into more of what that is. First thought to occur in North America. 1922, first confirmed case in North America of this hemorrhagic septicemia, and the bacteria isolated from that outbreak was eventually used for challenge studies and a lot of research that occurred in a bison herd in Yellowstone. Um, then in the 1950s and 1960s, I really think this was kind of the heyday for a lot of research on Pasteurella maltosida, with Namioka and Carter initially describing some capsular typing systems. There are 5 different capsular types of Pasteurella maltosida. So these two independently described some different methods for potentially typing these. The predominating system was developed by G.R. Carter. And then in the 60s, Carter and Holmes ended up attempting to characterize the different capsular types of Pasteurella maltosida isolated from human infections. So it was recognized at that time as potentially a zoonotic illness. So they talked about the different capsular types and potential animal hosts. And at the time, capsular types A and D were most frequently identified, but and a lot of non-typable strains were noticed at that time, but no type F strains had been described at that time either. So more than likely, that was what was going on. In the 1970s and 1980s, they ended up coming up with serotypic methods. So identifying or characterizing that somatic antigen. In 1987, then was when that type F strain, so more than 30 years from the original capsular typing methods being described, that final 5th capsular type was described. And it really is an important human pathogen. I think we've all probably run into it in the lab. It can seem kind of innocuous because it tends to be very susceptible to like the beta lactams. Acquired resistance is pretty rare for those. But an article in 2022 noted that more than 330 case studies were on human patients with Pasteurella multocida infections. So it is a pretty important cause of disease. Of course, I'm sure there are more in the literature since that article was written. But yeah, very, very significant in terms of it affects all animal species. Multocida literally translates out to killer of many. And of course, pretty much any animal species and humans could be impacted. And something that really struck me as I was going through old literature, I feel like the people that are interested in Pasteurella multocida tend to be very passionate about it. And how many bacteria have you heard as being described as enigmatic or fascinating in like a literature review? That popped up multiple times. I really appreciated that about the bacteria.
Wow, that's such a great historical overview and a lot of things that I wasn't aware of. So definitely thank you for that. And so one thing, and then as you touch on it, when right, Pastorella, if you're a medical lab scientist and you work with cultures, at some point in time, I'm sure you have seen it. And it's typically, right, you see that typical presentation where maybe you get it in a wound. You can also have all of the cases where then maybe spreads out and you can have it like in bacteremia or something like that. But typically the most classic one is you're sitting working on a wound culture and you see it there. And then you always check the history and you see, okay, yeah, dog bite, cat bite, depending on the species, like in the most exciting one for me was Pasteurella aerogenes and history, pig bites. Like, wow, in Orlando. That was definitely very fascinating. But maybe, as someone that I don't have any pets and I haven't worked with veterinary microbiology, but besides that, I don't think much about it and the way that I know that, it's part of the commensal, in some organisms. But beyond that, thinking about diseases in animals, I haven't given much thought. So maybe if you can talk to the audience and tell them more about Pasteurella multocida and disease in animals.
Yeah. So actually, and this was shocking to me, I guess, when I moved over to vet med, but of course, there's bovine respiratory disease complex. So that's primarily caused by bacteria and viruses, with most of the bacteria belonging to those Pasteurelliaceae family. So there's that Mannheimia hemolytica, Pasteurella multocida, and Histophilus somni. And globally, it's estimated that this respiratory illness costs over $1 billion. Just because of the expense of managing it with antimicrobial treatment and death loss of animals. So it is a very significant cause of respiratory disease in cattle. Um, in cats and dogs, you can get respiratory, and often those bite wound infections might be more of like the Pasteurella canis or some of those more minor species of Pasteurella. In rabbits, it sounds super cute, but they get what is commonly termed as snuffles. It's a respiratory illness in rabbits. Goats, sheep, even elephants and other ruminants can wind up with it. I'm sure any animal species has it could be described as having an infection with Pasteurella multocida, but similar to humans, would often expect it. So if the animal is stressed or immunocompromised, or if you have those bite wounds, um, so pretty common there. And of course, in humans, we realize that we'll often see them, as you had mentioned, in those skin and soft tissue infections, potentially more invasive infections and respiratory illnesses. And you'll usually see those in the young, elderly, or immunocompromised. But of course, a bite wound, I think that's pretty commonly associated with those cats. So not infrequent in immunocompetent competent individuals.
Yeah, and I think for some reason, I mean, it's just something that making that connection, and up to this day, I have seen many, and I don't know, it just you see it growing on the blade, and you're like, let me check that history, see if I see that bite. Like you're always making that connection, and that more than likely that's where it's coming from. But it's always, I don't know, it's just I don't know how else to say, it's kind of like fascinating, and so we always go back and check. Um, So right, so you talk about about the capsular types of Pasteurella multocida. You mentioned some examples, and you also mentioned hemorrhagic fever. So maybe if you can go over those, maybe to expand on the types and animals associated with each type and things like that.
Yeah, so there are five different capsular types. There's type A, B, D, E, and F. Type A would be the most commonly seen capsular type in humans for sure from several different papers, but also the most common for fowl cholera, which I forgot to mention in those animal diseases recap, but also that bovine respiratory disease, and then often in other ruminants like sheep and goats. Capsular types B and E cause that hemorrhagic septicemia. And that's actually a really problematic disease primarily in Asian and African countries or developing countries primarily. It causes really rapid death loss in cattle, and it's really characteristic in that it causes edema in the neck and like in the chest region or brisket region is how it's usually described. And then there's just prolific hemorrhage throughout the body for these animals. So it can be really devastating for those countries where there's a lot of dependence on cattle economically and, of course, for food. And then we have capsular type D, which is most commonly associated with pigs. So it causes what's called atrophic rhinitis, which essentially there's a toxin produced that ends up destroying the nasal turbinates in the pigs. But that type can also cause bovine respiratory disease. And then there's capsular type F, which is more minor. It is one of, from my experience, the more common capsular types seen from human infections. But I think it's probably pretty often seen in cats if we were to characterize more of those, but then also poultry. But really, any of those capsular types can jump around and cause disease in the different animal species and different areas, but those are the primary characteristics of those types.
I have worked in different labs, and I know that when it comes to a lot of times when it comes to things like like capsular typing and things like that, it sounds like maybe something that maybe will be done like by some sort of reference lab or like a state lab of sorts. So I don't think it's something that might be routinely available in the labs. Is that correct?
That is correct. Of course, it's an esoteric test method, so it wouldn't often be performed. Maintaining the antisera is really difficult. And I think as late as 2001, it was noted that maybe two labs in the world maintain that reference sera. And I know for sure, like our National Veterinary Services Lab in the US stopped routinely producing those. And of course, something we think of all the time is that diagnostic stewardship. If it's not actively going to help you treat something better or characterize the disease, is probably not practical. So where I see it being most practical is in cases when we're expecting like that hemorrhagic septicemia that would be unusual. Um, Primarily, but it is really interesting when you kind of want to get a little bit deeper into some of that epidemiology, and potentially there could be more virulence associated with some of those different capsular types. So it is very impractical for routine use. There's PCR methods for the capsular typing, and you can also use whole genome sequencing. There's actually some really great databases that will take the sequences and tell you the capsular types pretty rapidly. And it's really friendly for folks that don't have a lot of bioinformatics experience, like myself. Really great point and click option.
Yeah, I like that you mentioned. Yeah, I not recent, like last year, I took a bioinformatics class, and oh my goodness, I learned a lot, but definitely was a lot of information and very, the labs had like a lot of steps, and I enjoyed it, but definitely like a lot of information. It's something that I wasn't familiar with because when it comes to things like like sequencing, and as definitely it's trending now. Now you hear more research on it, and maybe some labs might be starting to bring it. But it's something that up to now, like routinely, us medical lab scientists, we haven't worked with in the lab, especially in microbiology. So we might not be familiar with some of those things. And then that brings me to MALDI-TOF. Side note, I definitely enjoy how the field has been changing, and from the traditional culture reading, then we started bringing MALDI-TOF. Now there's talks about, gene sequencing and things like that. I have done episodes about MALDI-TOF. I love some things about it. I do not love some things about it. But here, there's some model that was developed to use MALDI-TOF MS for differentiating hemorrhagic from non-hemorrhagic strains. Can you talk more about that?
Yeah, so that manuscript was part of a project that I wrote for one of my products for my doctoral project. Very much owe a debt of gratitude just off the top to doctors Stanger, Loy, Wynn, Clausen, and Roberts. Those are my co-authors on that paper. So this method was developed so that we'd have a rapid way of looking for those hemorrhagic septicemia strains in the lab. We see so many Pasteurella it would not be practical, as we'd mentioned, to actually do the capsular typing on those. So basically just using other models. There are some papers out about looking at different genotypes, say of that Mannheimia hemolytica, which is again that bovine pathogen, but also Haemophilus influenza type B. There's a paper that's described to be able to differentiate that from the other types. And so using that same thought process, tried to develop a MALDI-TOF MS method for capsular typing the different types of Pasteurella. Well, it became very apparent that there was a lot of overlap between the capsular types A, D, and F. Um, and so it made it really difficult to actually find a good biomarker. So those mass spectra peaks or the main spectral profiles that your MALDI-TOF is using to actually identify the different bacteria, you can actually find those different biomarkers and potentially associate it with genomic differences, and that's where you get into that nitty gritty with the different capsular types. So it was really hard to differentiate those alike strains, and so wound up looking to see if I combine the kinds that cause hemorrhagic septicemia and compare them against ones that do not. Is there a distinguishing peak that would actually help differentiate those? And fortunately, there was a mass spectral peak commonly found in all of the hemorrhagic septicemia strains was that 7.729 mass to charge ratio. There were some trickier strains that are, I think, emerging in North America and elsewhere in the world, but they might be our more predominating strains. And extra peak in there, an intensity difference between the non-hemorrhagic septicemia strains and the hemorrhagic septicemia strains really helped identify those further. So we can do it with a model and software that would do it automatically if you were to pull the spectra from your MALDI and put it through that. Or you can actually, I know on my research use only or the RUO Bruker model that I have, I'm able to actually look at it directly in the software to see if that peak is present or not. And seeing those peaks, I would then be able to decide, should I actually pursue sequencing or some other kind of characterization, because this is a reportable disease for animals in the United States. So it's one that we keep track of. So it would actually give us a tool. These strains, of course, if you've seen them in the lab, you might see ones that are more mucoid than others. Some will be a little bit more dry or shiny looking. But you can't just look at it and be like, that's a hemorrhagic septicemia strain. So it's just a really useful tool. It doesn't necessarily cost extra money because you can look at your direct spots that you're normally doing in the lab. You could potentially do your formic acid overlay, or you could do a full formic acid tube extraction, with that tube extraction being the most sensitive and producing the crispest peaks that you could possibly have. So just kind of a more integrated way to examine these capsular types.
It's always something interesting how, like I use the Vitek MS and when we talk about the formic acid, with the Vitek, it's only used for fungi, like if you have yeast. So when it comes to this model, are there any next steps for this, or that's about it, or?
My next dream would to use Fourier transform infrared microscopy or mass spectrometry, FTIR. My next goal would be to use FTIR to be able to actually get more granular examination of the different capsular types. I think that's really the only other way to go about it, for creating a better model that wouldn't be something genomic or sequencing. Um, but in the meantime, it would be additional typing potentially of these strains using that whole genome sequencing. I do, related to this project, have a couple of other manuscripts that'll be coming out. So we're describing potentially the first two cases of hemorrhagic septicemia in cattle in North America or in the United States since 1993. So it is very rarely described. And then we also have another manuscript, both just submitted, describing Pasteurella multocida strains that were collected from human patients. So describing those more granularly. So just some interesting things. We also typed those human strains using MALDI-TOF MS. And of course, as expected, most were those A, D, and F strains, or all of them, I should say. Um, that is something I don't think I mentioned. There have yet to be reported human cases of hemorrhagic septicemia, but I think that would potentially be more towards resource limitations or potentially just people not thinking of them being different, especially if you're in those developing countries and you have these other strains. So I think that would be really interesting is to actually find out from humans that are getting Pasteurella multocida infections in those endemic countries, are we actually seeing those strains? Is that something that's potentially impacting folks in those areas?
Well, I'm definitely looking forward to those publications and be able to read them. And when it comes to this one with the model, has this been published already that maybe the listeners can look at it?
Yes, actually, it just published in the Journal of Microbiological Methods, and it is titled Development of a MALDI-TOF MS Model for Differentiating Hemorrhagic Septicemia Causing Strains of Pasteurella multocida from Other Capsular Groups, if folks want to check that out.
Perfect. And I'll go ahead and put a link on the show notes. So if you want to check it out as you're listening to this episode, once you listen to it, if you want to go ahead and take a look at it, it's going to be there for you to access. Um, so right when and you told me you listened to it, and I did an episode, when I was starting the podcast about why I like it so much, and I also, and you mentioned this when you were in your introduction, but so it is also your favorite, right? So you also like it as much as I do. So can you tell the audience why is Pasteurella multocida your favorite organism?
Well, at this point, after spending so much of the last 2 years really researching this bug, it would be really hard not to love it. I think it's imprinted on my soul at this point. But as a young med tech, it was one of my favorites to get. Just really similar to you, you end up for me, you're flipping through your different culture plates. You see the growth on the blood and the chocolate agar, then nothing on CNA or MAC. And I know as a young tech, I was like, this seems like an E. coli, but it's weird that it's not growing on MAC. And you end up going through indole and oxidase, realizing those are both positive. And then it would click in my head. I should look at um, the record and see if there was animal contact of some sort. Um, Now knowing more about the literature, there's a lot more where we're seeing with those immunocompromised patients, just them having a pet and being really close and maybe cuddling. Maybe they're going through cancer treatments, they become colonized, and it can then be a problem. But of course, even just that interaction of the human animal bond then contributing to a disease. I also just love that it's really susceptible for antimicrobials. For some reason, that's a charming quirk for me on that. But yeah, it's just a great bug. It's predictable, but also has its unusual little quirks there too in the different colony types. So just kind of a fun bacterium for sure.
Indeed, and like I always tell the techs, if it's not growing on Mac, stop and think, do a gram stain. I have seen people go through the worst outcome, which is you're assuming that it's positive, and then you put it in an instrument, you get an ID, and so many things going wrong that way. So take your time, but it's still, and to this day, I know when I'm looking at all my plates, and sometimes from the get go, I see the McConkey that is not growing, and or I see gram stain, gram negative rods, and I see the two plates, it still makes me smile. It's like the same thing with Haemophilus influenza. When I look at that blood and I don't see anything, and then I flip that chocolate and those 10 colonies are fully grown in there, oh man, I don't know. I guess it brings the nerdiness in me. So, but definitely, wow, what a great episode. I'm learning so much about Pasteurella, and like I mentioned before, yeah, you don't maybe you might think about it in the terms of you see that wound in the lab and something like that, but all those things that you have brought to light about patient maybe is immunocompromised, you have a pet, you might get colonized with it, it really puts a lot of things in perspective. And of course, with all the antimicrobial resistance that we're seeing, it's always nice when you look at an organism, the profile is nice and susceptible. It definitely, it's a good thing. So, well, Kelli, as we are coming to the end of the episode, is there anything else that you want to tell the audience?
I just really hope you gained an extra appreciation for this cool bug. It is such an interesting pathogen and it has such an interesting history. That was another thing I absolutely loved about researching it. There's so many papers and debates. I feel like I know the people that researched it in the past to an extent. It's one that I feel like has been built upon many, many times over the decades since it was first described by Pasteur. So it's really fun to be a part of that legacy. Um, I just hope you can join in that passion and maybe appreciate it a little more the next time you isolate it in your bench work.
Well, thank you for that. And for the listeners, if you haven't, as I said before, if you haven't listened to the episode about veterinary microbiology, it was recently re-released as part of the Throwback Thursday series. So if you have your catalog set up on the order of release, you should be able to see it kind of right at the top. So check it out. Well, Kelli, once again, it was so great. I enjoyed the first time that we talked. I talked to you and Sarah, and definitely I truly had a great time recording this. So thank you so much for taking the time to be in Let's Talk Micro.
Thanks for having me back. It was great.
My pleasure. And that, my dear audience, it's the end of this episode. I hope you enjoy learning more about Pasteurella, like capsular types, a little bit of a historical overview, and more. As always, I enjoy sharing this information with you. And so great, having Dr. Maddock in and talking about Pasteurella, my favorite organism. So, as always, continue bringing that passion to what you do. It's so important. You do such great work. And stay tuned. Great things coming your way. Thank you so much for the support. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.